Bismillahirrahmanirrahim. Today we are going to learn about the internal ear and mechanism of hearing. To know about the internal ear and mechanism of hearing, we are going to the previous lecture. In the previous lecture, we have learned about the external ear and middle ear, and today we will learn about the internal ear. The external ear ranges from pinna to the tympanic membrane, and in the middle ear consists of all those structures which are enclosed in the tympanic cavity. The sound waves come across the concha of the external ear and go through the external acoustics meatus and strike with the tympanic membrane. The tympanic membrane start vibration and the vibration is transferred to the malleus. From the malleus, the vibration is transferred to the incus. From the incus, the vibration is transferred to the stapes. From the stapes, the vibration is transferred to the oval window, and after transferring to the oval window, the oval window transfer the vibration to the internal ear. As we focus on this diagram, so the internal ear start from this oval window, and the vibration which is transferred from the stape to the oval window is transferred from this oval window to the perilimb of this internal ear. Perilimb is a fluid which is present inside the internal ear in this area. Going to the detail, the internal ear consists of this structure, snail-like structure. This is called cochlea. This structure is called vestibule, and this structure is called semicircular canal. So three structures are there. I have written their names. The cochlea, the vestibule, and semicircular canals are there. The cochlea is responsible for hearing, while the vestibule and semicircular canals are responsible for equilibrium. This structure is responsible for equilibrium, while this structure is responsible for hearing. We will learn about this structure and the whole mechanism of hearing in this lecture. So going to the detail, you can see over here that the oval window open into canal. Open the oval window opens into an area. This area, this is called cochlea, and the cochlea is having three pathways: the superior pathway, the middle pathway, and inferior pathway. The superior pathway is called, this pathway is called scala vestibuli. This is called vestibular duct or scala vestibuli. The middle one is called scala media or cochlear duct. The lower one, the inferior one, is called scala tympani, or you can also call it tympanic duct. So three ducts are there: vestibular duct, cochlear duct, and tympanic duct, or scala vestibuli, scala media, and scala tympani. So what happens to these pathways? The scala vestibuli over here, the scala vestibuli is uh, connected with this pathway at the end. I'm going to show you this pathway as you go farther. This pathway over here, this pathway is connected with the scala tympani. Scala vestibuli is connected with the scala tympani, and this opening, the opening through which this pathway is connected with this pathway, this opening is called helicotrema. Helicotrema is the opening which connect the scala vestibuli with scala tympani. Now coming towards the membrane which divide this or which separate the scala vestibuli from scala media. This green color membrane, this membrane is called vestibular membrane. This membrane is called vestibular membrane. Vestibular membranes separate the scala vestibuli from scala media. Now this blue color membrane, this membrane is called basilar membrane. Basilar membrane actually separate the scala media from scala tympani. This the blue color membrane, the basilar membrane, separate the scala media from scala tympani. So the fluid which is present in the scala vestibuli is called perilimb. As this duct is connected with the lower duct, with the scala tympani, so both of these contain the perilimb. Perilimb is also over here. The lower duct, the scala tympani, and the scala vestibuli, both of these contain the perilimb. while the scala media, the middle duct, this contain the endolymph. Endolymph is having just like this composition, just like the cerebrospinal fluid, and this endolymph is present inside the cochlear duct. This is also called cochlear duct. The scala media is also called cochlear duct. Now coming toward the next slide. In this slide we can see that the cochlear duct also consists of another membrane above the basilar membrane. This is the basilar membrane, and above the basilar membrane there is a fine, a delicate membrane, the red color membrane you can see over here. This membrane is called tectorial membrane. This is very important membrane. Tectorial membrane, a small or delicate membrane which is present above the basilar membrane inside the cochlear duct. I will show you the function of this membrane in the next slides. Now coming toward the next slide. In this slide we can see that the basilar membrane and the tectorial membrane also consist of cells over here. In between the basilar membrane and the tectorial membrane there are cells, and these cells are called hair cells. These cells are called hair cells. These are actually the hearing receptors. These are actually the hearing receptors which are responsible for hearing. These contain hair-like projection, the cilia. That is why these are called hair cells, and this is responsible for listening, for hearing. So now coming towards the next slide. Uh, it will be better to compare this slide with that one. You will be able to see it very clearly. Let's suppose this one. See this one. This is the cochlea, the snail-like structure, and if we get the cross section of this cochlea, the cross section of this area, what will we get? We will get this cross section. We will get this one. This is the cross section. This is the scala vestibuli, and scala vestibuli is represented over here. This is scala vestibuli, which contain the perilymph. This membrane is called vestibular membrane, which separate the scala vestibuli from the scala media. So this is the scala media. This partition is the scala media, and scala media is separated from scala vestibuli with the help of vestibular membrane, and this is the vestibular membrane which divide or which separate the scala vestibuli from the scala media. This is basilar, this is vestibular membrane, which is represented in green color over here and also green color over here. Now the third one, this layer, this is the basilar membrane. The blue color membrane is the basilar membrane. Over here, the basilar membrane is here. This basilar membrane separate the scala media from scala tympani. So the lower partition over here, this is the scala tympani. This is the scala tympani, and this is the scala media. So the scala media and the scala tympani is separated with the help of what? With the help of basilar membrane. Over here the basilar membrane is this one, and over here the basilar membrane is this one. Now you can see the red color, the tectorial membrane, and tectorial membrane is also over here. This red color tectorial membrane is inside the cochlear duct above the basilar membrane, and in between the basilar membrane and tectorial membrane there are fine cells. These cells are called hair cells, as I have represented in dots. These are the hair cells. So these are the hair cells. What are these hair cells doing? Why these are called hair cells? Because these hair cells contain the cilia-like structure, which are called stereocilia, and these stereocilia are connected with the tectorial membrane. These are connected with the tectorial membrane. Tectorial membrane is placed upon these hair cells. Okay, and these, due to these hair-like structure, these are called hair cells, and these hair cells are connected at the base with the cochlear neurons, with the dendrites of the cochlear neuron. These are the dendrites of the cochlear neuron, and the dendrites of the cochlear neuron are connected with the cell bodies of these dendrites. So the cell bodies are concentrated over here making the spiral ganglia. This is called spiral ganglia, and from the spiral ganglia, the nerve arises, and this nerve is called cochlear nerve. This is called cochlear nerve. So what happens to the sound wave, and what happens uh, to the vibration of sound which comes across the whole ear? I'm going to show you in detail. The For example, focus on this diagram. In this diagram we can see that the sound waves which go through the external acoustics meatus is transferred to the tympanic membrane. From the tympanic membrane, this sound waves is transferred to the malleus. From the malleus, this is transferred to the incus. From the incus, this is transferred to the stapes. From the stapes, vibration is transferred to the oval window. From the oval window, this vibration is transferred to the perilimb. Now coming towards this perilimb, the vibration is transferred to the perilimb. From the perilimb, the vibration is transferred throughout this whole semicircular, throughout this canal. This goes through all the way up to the helicotrema, and at the point of helicotrema, it bounces back towards the lower, the scala tympani, and it goes once again away to reach this area, to reach the oval, to reach the round window. So from the oval window, this vibration goes through all the way to helicotrema and get back to the round window. This is one way up transmission up transmission up what up vibration. And what is the function of this round window just is act as absorbent. It absorb that vibration. Now this sound waves, or this wave, also goes through this membrane. This sound wave also goes through this membrane, this vestibular membrane. This sound wave is transferred to vestibular membrane, and vestibular membrane start vibration. From the vestibular membrane, this is transferred to the endolimb. From the endolimb, this vibration is transferred to the to the tectorial membrane, and from the tectorial membrane, this vibration is transferred, as tectorial membrane is connected with the hair cell. So tectorial membrane uh, transfer this vibration to the cilia of these cells. And when the cilia of these cells get vibration, they bend, and bending of these cilia open the potassium channel in these cells. And by opening the potassium channels of these cells get action potential. And when this get action potential, the action potential, due to action potential, this releases the glutamate, and glutamate is secreted towards these neuron nerve ending, and these nerve ending get the action potential, get the impulse towards the cell body, and this get the potential towards the brain. Now coming toward the detail, what happens normally, uh, in this slide we can see that this is a hair cell. This is a hair cell, and this hair cell is having cilia. At the apex, this contains cilia, and at the base of this hair cell, the nerve ending of the dendrite of the cochlear neurons are there. So these hair cells contain one group of cilia which are very long cilia, and these all of these cilia are called stereocilia. All of these cilia are called stereocilia, and the stereocilia, one of the stereocilia um, which is very long, and that is called group of stereocilia which is long, which is having very high length, that is called kinocilium. It is called kinocilium, and this kinocilium is sometimes considered as not motile. This is not moving. All of these can move, but this will not move. Sometimes this can also move, but this depend upon the situation to situation. Now I am going to show you in detail. These stereocilia contain, I'm going to maximize this photo. You can see over here that at the tip of each stereocilia there are a large number of channels. I have made one channel, but there are a large number of potassium channels at the tip of each stereocilia. There are large number of potassium channels. So these, what is the function of these potassium channels? These potassium channels are connected with the spring-like protein that is called tip-link protein. Tip-link protein. Each potassium channel is connected with the tip-link protein, and one end of this tip-link protein is connected with the lateral side of the stereocilia. The other side of this tip-link protein is connected uh, with this potassium channel. In next diagram, in this diagram, you can see this is one stereocilia and this is another stereocilia. So this is a tip link protein and this is the potassium channel. So this potassium channel is having a lid like structure. This lid is connected with the tip link protein, and tip link protein is a spring like structure. This tip link protein, this end of tip link protein is connected with the tip of this potassium channel, and the another end of this tip link protein is connected with the lateral side of stereocilia. So what happens normally when this cilia contain tectorial membrane. Upon this cilia, upon each of these cilia, there is tectorial membrane. When tectorial membrane start vibration, these are bent to one side. To this side, these are bent. These stereocilia will bend to one side. While bending to one side, if this stereocilia bend to this side, what will happen? It will open this channel by bending of this cilia. This potassium channel will open. How will this open? Because this is connected with this potassium channel. If this get open, this spring will become strained. This spring will become extended. While being extended, what will happen? It will open this channel, and when it opens the channel, the potassium which are present in the endolymph contain large amount of potassium. So this potassium will get entered into the cilia. This potassium will get entered into the stereocilia. So when this potassium get entered inside the cilia, what will happen inside the stereocilia? What will happen? We will see it in this diagram that this potassium, the potassium is going to move inward. When this potassium is going to move inward during the bending of these stereocilia, this potassium is going to go move inward. By moving inward, what will happen? This will increase the positivity of this cells. This cell, this receptor cell, or this cell, the positivity or positive ions will increase inside this hair cell. When the positivity of this hair cell is increased inside and the negativity is increased outside, what will happen? The An action potential will be generated on its cell membrane. The action potential, as I have told you in the previous lecture about the node of Rinveer, in that lecture, that when for each cell there is a threshold for the positive ion to get entered. When a specific threshold of that positive ion get entered into the cell, that can arouse the action potential. So the action potential is initiated over here. When it started, the action potential over here, you can see that the positive ion is inside and the negative outside. So the movement of positive ion through the membrane, this actually shows the action potential. So action potential is transferred through the cell membrane, through this membrane, uh, all the way toward the base. And when this is going toward the base, at the base of this hair cell, there are calcium channels. The calcium channels opens due to the action potential. When the action potential come across these calcium channel, the calcium channel get open. So by opening of the calcium channel, the calcium from the outside environment is going to be transferred to the inside environment. From the influx of calcium will do what? It will increase the calcium concentration inside the cell, and by increasing the calcium concentration inside the cell, the granulation of these glutamate neurotransmitter will take place. These neurotransmitter which are present inside the vesicle, the glutamate neurotransmitter, will be secreted in the synaptic cleft. When the glutamate is secreted in the synaptic cleft, what will happen? These glutamate will bind with the receptor on these nerve terminal, and when it bind with the receptor, it will start an action potential, and this action potential will be transferred towards the cochlear neuron, towards the cell body of this cochlear neuron. and when this action potential is transferred toward the cell body of, this is the cell body of this cochlear neuron. When this is transferred to cell body of cochlear neuron, it will goes all the way through the axon toward the brain, and this nerve is called, this is called cochlear nerve. What will happen over here, you can see that these are this is the tectorial membrane, this is the basilar membrane, and these are the hair cell. From the base of each hair cell, there arise the neuronal terminal. You can see these are the neuron which arise from these are the cochlear neuron. The cochlear neuron arise from the hair cell, and the cochlear neuron which arise from the hair cell gave rise to these cochlear neuron make a nerve, and that nerve is called cochlear nerve. This is the cochlear nerve. A large number of cochlear neurons combine together to make the cochlear nerve. So the vestibular nerve also arises from the vestibular apparatus. These are the vestibular nerve, and this is the vestibular nerve. The vestibular nerve combined with the cochlear nerve to make the vestibulo-cochlear nerve. This is very important nerve. This is the vestibulo-cochlear nerve, and this vestibulo-cochlear nerve go through internal acoustics meatus into the brain. This vestibular cochlear nerve go through internal acoustics meatus into the brain. As I have told you earlier that the this in this diagram you can see that this is the external acoustics meatus. This is the external acoustics meatus, while this is the internal acoustics meatus. What is the difference between the internal acoustics meatus and internal acoustics meatus? The external acoustics meatus is doing what? This is the external acoustics meatus. The external acoustics meatus get the sound vibration, the sound frequency through the eardrum. This is the external acoustics meatus, and the internal acoustics meatus is the inside the internal ear. This is the internal acoustics meatus, which give the connection to brain through the internal acoustics meatus through this hole. This is a hole. This is also a hole-like structure. This is also a hole, a tube-like structure. Through this internal acoustics meatus, the vestibulocochlear nerve go to the brain, and when it goes to the brain, what will happen? This is the vestibulocochlear neuron or vestibulocochlear nerve. So this vestibulocochlear nerve will go towards the medulla oblongata. This is medulla oblongata, and the nucleus inside the medulla oblongata, that is called cochlear nuclei or cochlear nucleus. This is cochlear nucleus. It will go toward the cochlear nucleus. This is the first order neuron. This nuclei will give two types of axon, two types of neuron. One will go directly to the pons, and the other will go to the opposite side of the cone. Similarly, the case from this side, one will go on this side and one will go on this side. So these are called olivary nucleus. These olivary nucleus inside the pond. Olivary nucleus is just like the olive, which we eat, the olive, which is very useful. So from the olivary nucleus, the third order neuron will goes toward the midbrain, and from the midbrain is having a nucleus that is called colliculus. So colliculus, from the colliculus, the fourth order neuron will goes toward the thalamus. This is the median geniculate nucleus of the thalamus. The median geniculate nucleus of the thalamus will receive the fourth order neuron, and from the medial geniculate nucleus, the auditory radiation. These are called auditory radiation. The auditory radiation will start moving towards the primary auditory cortex. The primary auditory cortex is present in the cerebrum, in the cerebral cortex. In which part of the cerebral cortex? In the temporal area of the cerebral cortex. In which part of the temporal area? In the lower operculum of the temporal lobe of the cerebral cortex. So in the lower operculum of the temporal lobe of the cerebral cortex, the auditory nucleus is there, the auditory area is there, and this auditory cortex is responsible for the reception and interpretation of various sounds, of various voices. One more thing which is very important, when the sound vibration go through this wool window into the uh, perilimb of the internal ear. So this area, this is called cochlear duct. The cochlear duct is very strong and made up of very strong fibers in the near this wool and round window. As it goes onward, this is very weak at this side. This is very soft and very weak at this side. The fibers are very soft and very weak, so the very low frequency sounds are being detected by the helicotrema side, by this side. The hair is helicotrema, so near the helicotrema the 20 hertz frequency sound can be detected very easily, while near the oval window the 20,000 hertz sound can be detected very easily, because this is very difficult to vibrate this area. The vibration of this area is very much easy, and the vibration of this area is very much difficult. So this side detect the sound frequency up to 20 hertz, and this are above 20 hertz, and this area detect the sound frequency up to 20,000 hertz. And the normal voices which we observe and which we detect in day-to-day life, that ranges from 1000 up to 5000 hertz, and that is detected by the middle area of this cochlear duct. One more thing very important, the hair cells inside this area, this is the cochlear duct, and inside this cochlear duct, this arrangement, this organ like structure, this is called organ of Corti. This organ is called organ of Corti, which contain the tectorial membrane, which contain the hair cell, which contain the supporting cell, which contain these nerve terminal. So these are called organ of Corti. This organ of Corti contain the inner hair cell and the outer hair cell. The inner hair cell is only having one row. As I have made one hair cell over here, it means that over here the whole length contain only one row of cell. This is the inner hair cell. This is only one row of hair cell, and these are the outer hair cells. And outer hair cells uh, are having three rows of these hair cells, and each of these hair cells contain up to 100 stereocilia. And these cells, one thing more thing very important, that the inner hair cells are connected 90% sensory neuron and 10% motor neuron, while the outer hair cells are connected with the 90% motor neuron and 10% sensory neuron. And what is the function of motor neuron, by the way, over here? The function of the sensory neuron is to get the sensory information of sound and toward the brain, while the function of the motor neuron is to modify the sensitivity of these cells. The function of the motor neuron is to modify the sensitivity of all these cells. So these cells are having, these three rows of cells are having, the outer rows of cells are having the 90% motor neuron, and these cells are having 90% sensory neuron. So to modify the sensitivity to various sounds, these are very much sensitive.